Hey everybody, so today we're going to be reviewing the chemistry primer for cellular respiration, right? And so in order to understand cellular respiration, we first need to understand enzymes and how they behave, okay? So what is an enzyme, right? So enzymes are basically these proteins that make reactions happen faster, right? They're like a kind of motor. So if we look at this not that picture, at this picture right here, right? So these two pictures show us how enzymes work. So in the body, you have these molecules called substrates, and these substrates are what go into an enzyme and bond to this thing called the active site to let the enzyme do its work. Okay, and so enzymes can do one of two things. They can either combine two substrates, or they can take one substrate and split it into two. So first, let's look at the case where the enzyme is making two substrates bond together. So if we didn't have enzymes, then the two substrates would have to like basically collide with the correct speed, at the correct orientation, with the correct amount of energy for them to be combined into one substrate, and that's extremely rare, like extremely unlikely, right? And so what an enzyme does is it recreates these exact conditions for them to bond. So it'll reorient them and it'll like push them together to form one substrate, right? So if you think of like a Lego box, right? If you had a bunch of little pieces in this Lego box and you dump it onto the floor, what are the chances that two pieces are going to become clipped together? They're going to become one piece. It's near never. It's like not going to happen unless you're very lucky, right? And so, but if you think of your hand pushing them together, right, that's basically what the enzyme does. It's guiding them into the proper orientation for them to be pushed together. Okay, but the enzyme can also break apart a substrate. It can take in a substrate and it can break them apart into their two separate components. Okay, so that's basically what an enzyme does, right? And also, one more thing to note is when these things happen spontaneously, meaning there's no enzyme to help it out. They happen randomly. It requires a lot of energy because it's so unlikely. So that requires a lot of energy with a lot of these molecules for even one to occur. But when you catalyze it or when you use an enzyme to do this, it requires a lot less energy because you don't need many pieces and you don't need to use a lot of energy with like those pieces having to move and such. And so it'll orient everything properly. So it'll save you a lot of energy. Okay. So let's look at these graphs now. And so these graphs show basically what we just discussed about enzyme and non-enzyme and the amounts of energy. Except in this case, we're talking about how fast a reaction is going to happen. So if you notice these blue lines, like right here and right here, those lines are the non-enzyme, and these red lines along this and here are with an enzyme. And so our y-axis, or our vertical axis here, shows how fast the reaction is happening, the rate of the reaction, okay? And so when the reaction is going at 100%, like the maximum speed it can go, it's called Vmax, or the maximum velocity, okay? And then another important measurement is 1/2 Vmax, or 50% of the rate of reaction, half of the maximum velocity, okay? So if you notice, with the non-enzyme, the blue line, it's completely straight and it stops after a point. So on the x-axis, if you notice, it's talking about the concentration of the substrate. So anywhere from 0% to 100%. So at 100% a substrate concentration, it's only reaching around 1/2 Vmax. It can't go above that, right? But with the enzyme orienting everything properly so that they either come together or are split apart, right? Notice that it actually reaches Vmax, reaches the peak performance at around like 30% of substrate concentration, right? So this really shows how easily enzymes can make these reactions happen. They make them happen a lot faster and a lot easier with less energy. Okay? Whoa. And so this graph is showing basically the same thing, right? So at 1/2 Vmax, this non-enzyme catalyzed reaction, so basically this happened spontaneously, it only reached about 50% at maximum capacity. But the enzyme was able to reach maximum velocity at like 30-40% of substrate concentration. Okay? So if you have enzymes, you don't need as much substrate. But if you don't have an enzyme, you're going to need a lot of substrate to make sure it happens. Okay? And so that's basically a lot about enzymes, right? And so enzymes are extremely special because they are required, they're critical for many important biological processes, whether it be digesting food or producing energy that your body needs to move around and do whatever it needs to do. Okay? So now we also have to talk about one other thing called inhibitors. And so inhibitors are, as you may have guessed, inhibit the reaction or they stop the reaction. So there are two types of inhibitors, okay? So I'm going to pull up an animation to let this be a little clearer, okay? So two types of inhibition. One is competitive and the other is non-competitive, okay? So, in this, so here, in competitive inhibition, you have the one active site, right? So the inhibitor and the substrate are racing toward that active site. So whoever gets there first gets to keep it. So in this case, the substrate got to it first, and so the inhibitor has to go away. But in this case, the inhibitor gets to it first, and the substrate can't bond, and so it goes away. Okay? But now there's also a second one. It's called allosteric competitive inhibition. So the principle is still the same. They're still basically running a race toward their active site, except they have two different end goals, two different finish lines, right? So there's one active site for the substrate, and there's one active site for the inhibitor, right? So they're still racing to see who gets there first. But if you get there first, then the other site will no longer work. So let's look at this. So in this case, the inhibitor gets there first. And notice, right up here, the shape has changed. So now this substrate can no longer bond, okay? And so that's basically the point of allosteric. If one thing bonds, it'll change the bonding site. It'll change the shape of that bonding site so that the other thing cannot bond. So in this case, it had changed the bonding site of the substrate active site, so the substrate cannot bond. But if the substrate gets there, it'll change the shape of this, and the inhibitor cannot bind. So then we also have a second type, which is called non-competitive inhibition. So non-competitive inhibition is there's no race. The substrate can get there to start the reaction, but the inhibitor can go at any time and stop it, right? So if the substrate gets there first, right, the inhibitor can still get there. It can still bond to that second bonding site, to its bonding site, and stop the reaction, no matter where the reaction is. So the inhibitor doesn't care who gets there first. If it wants to stop it, it'll go stop it whenever it wants. There's no racing to get there, okay? And so those are basically the two types of inhibition. So competitive, the racing, and non-competitive, the inhibitor doesn't care. It can stop it whenever it wants, okay? So this is basically a chart of how the different types of inhibition change the enzyme kinetics. So basically the amount of critical mass needed to reach 1/2 Vmax and the Vmax, right? So competitive, right? In competitive inhibition, they're both racing toward the active site. The Vmax doesn't change, but you need more of the critical mass, right? Because now they're both racing. So if you have more of something, it's more likely to reach there. So you need more substrate if you want the reaction to happen. But if you have non-competitive, where they're binding at different places, right, then there's a decrease in Vmax, because a lot of the times the reaction will occur, but it'll stop before it can reach Vmax, or before it can reach a certain point, because the inhibitor binds and stops it. But there's no change in Km because you don't need more substrate. It doesn't matter how much substrate you have. An inhibitor can bond at any time. So there's no benefit of having more substrate, right? So we look at these graphs. Here in uncompetitive, so if you notice, these lines are red and blue lines are practically the same. And so this blue line is showing how Vmax has been decreased, because no matter how much substrate concentration there is, which is the x-axis, this horizontal line, the Vmax is decreased because the inhibitor is able to bond at any time and stop it. Okay? You can stop at any time. So it's in almost all cases, or not almost all, in practically every case, it's never going to reach Vmax, right? But in competitive, right? You see here, this blue line this straight blue line. So as your substrate concentration is increasing, the rate of reaction is happening faster, because since you have more substrate racing for that one spot, so it's more likely to reach that spot than the inhibitor. Okay, so that was everything you really need to know about enzyme kinetics. So enzymes can break apart a substrate, or they can make a new substrate out of two old substrate pieces, and they orient everything so that it happens properly. So it makes things happen faster and easier. Okay, faster and easier. As you can see in these graphs, right? So the enzyme is reaching Vmax, the 100%, the maximum rate at which reactions can happen much faster. Okay, so remember, if you guys have any question, feel free to contact us at our email, or you can ask it after lessons every Friday. Okay, hope to see you soon.